Look at the split screen diagram in front of you. On the left side, you have raw genetic data, jagged, unaligned rows of text. But on the right, that same text shifts into a structured, color-coded grid where the matching letters lock into clean vertical columns. To get from that raw text to a clean structural alignment, researchers rely on Clustal Omega. It is a fast, scalable tool used to process 3 or more sequences to help determine their evolutionary relationships. To follow along with this tutorial, you only need 3 things: a command line interface, a basic text editor, and your raw sequence data. If you have never used a command line before, do not worry. We will go through the exact commands you need to type step by step. Mastering this alignment process transforms raw, disparate sequences into a standardized format required for downstream phylogenetic trees or functional analyses. This infographic outlines your 3 main installation paths, which depend on your specific operating system. If you are working on a Mac, you can open your terminal and use Homebrew by typing brew install Clustal Omega. On a Linux machine, use your distribution's package manager. For Ubuntu or Debian, you will type sudo apt-get install Clustalo. And for Windows, simply visit the Clustal Omega website, download the executable file, and follow the standard installation prompts. Before you begin any actual work, there is one critical step. You must verify that your computer actually recognizes the software you just installed. Open your command prompt or terminal window. Type out Clustalo-H and press Enter. You should immediately see a dense block of help menu text populate the screen. Forcing the system to display this help menu confirms your installation was successful. Checking this now prevents confusing silent errors later when you attempt to align your actual data. Step 2 is preparing your input sequences. You will do this inside a basic text editor. Every sequence must follow the strict FASTA format, requiring a greater than symbol followed by the sequence name on the first line. The DNA, RNA, or protein sequence must begin on the next line, completely uninterrupted by spaces or empty rows. Once your sequences are stacked properly, save the document as input.fasta. Explicitly check the file to ensure there are no hidden spaces or special characters hiding at the ends of your lines. Command line programs do not give you friendly graphical warning pop-ups when something is typed incorrectly. Absolute adherence to these FASTA formatting rules is the only way to prevent the algorithm from crashing when reading your file. We are now ready for step 3, execution. Before running the alignment, your terminal must be pointed directly at the exact folder containing your input file. Use the cd command in your terminal to change directories, navigating to your active working folder. This command string runs the alignment in 4 parts. First, the -i flag reads your input.fasta file. Next, the -o flag names your finished output, output.aln. Finally, the --outfimp flag forces the output into a readable Clustal format. Press Enter. The terminal will pause for a brief moment as it processes the sequences, and then your new output file will appear directly in the folder. That covers basic execution. Step 4 introduces an advanced optimization technique for complex or very large datasets. By default, the program runs one single pass. You can append --iter equals followed by a number to the end of your command string to force the algorithm to run multiple passes over the data. Running multiple iterations leverages Clustal Omega's progressive alignment algorithm. It recalculates the guide tree with each pass, increasing the mathematical accuracy of your results. To review your work, open that newly generated .aln file in your text editor. Look at this alignment output file. Notice the newly inserted dash characters padding the text. These physically push the sequences left and right to force vertical alignment. Those gaps are mathematically calculated. They align conserved base pairs across your different species or samples, so they sit in the exact same column. You can then take your newly aligned output file and directly import it into visualization software like JALView or BioEdit. Transforming raw text into these clear visual motifs empowers researchers to identify structural patterns, predict gene functions, and map their evolutionary history.